Google is your best friend. My favorite like scientist Google story. I had this advisor that was on my my committee for my thesis with my master's degree, and I needed to go and talk to him. And so like I was talking to him. I sat down in his office, and he said, "Oh, I've got an important phone call here." As the phone rings, he picks it up, and he spends about 25 minutes talking to this guy about some process that he was doing. And while he was doing that, he was typing into Google to figure out how to actually do the process. So he was paid like, I don't know, a couple $100 for his 25 minutes of advising, and all he was doing is hammering away on Google. So again, if you guys end up going into a profession where you use computers a lot, those sorts of things, again, if you're tech savvy, if you can Google something, you can get a long ways with information. All right, so here we go. And so what we're going to have is we're going to have 26 samples, okay? Our microcentrifuges have slots for 24 samples. So we're going to divide up the room. If you guys are on this half, you guys are going to come up and grab one of these tubes. Let's see here. How do we have them numbered? Just rearrange them a little bit here. Okay, if you guys are on this half, you guys are going to grab a tube from the front portion of this block. If you guys are on this half of the room, you'll grab a tube from the back portion of this half. So there should be 1 through 13 taken care of by all of you guys, and there should be a 1 through 13 taken care of by all of you guys. All right? So there's 13 total samples. We know that we had 13 people in the family. You guys did the pedigree on that, and so we're gonna divide it up. Now, if you notice, what do we have? We have 4 and 4 and 4, which would only actually give us 12 people, right? So somebody will do 2 samples, or we can have our student teacher here, our assistant, do 1 sample also. And then you guys will have 13 total samples done. So let's go ahead and see if we can get that done. Grab a sample, and we'll take it over to the microcentrifuges. They're not organized in any fashion.
So I could say, GoPro, take a photo. GoPro, take a photo. Should take a photo, I don't know. Maybe it's not on. Oh, I need to wipe the lenses.
Seconds, 72 we run for a minute. We're gonna only repeat this 20 times.
Okay.
So it'll go through it once, and then 19 times it'll go back to that first step. That's what that's saying. Whoa. Let's cancel that.
So is this a therma?
It'll be a thermal cycler, but we're not quite ready for it. Okay. So this is a program that we want to run when it's time. NP for Niemann pick.
Okay.
Okay. So what you're gonna do when it's time to run, you'll hit run. You'll select Niemann pick. You'll do 25 microliters, and for right now, we'll leave it on view mode so that people can see the different pieces, right?
Okay.
So,
Then I'll click cancel and then go into it.
Yeah, you'll click cancel and then go into run. So you can set them in any order, whatever organization you want. If you want to do like north on one side and south on the other, that's fine. Okay, guys, we've got somebody who knows how to use the thermal cycler now. So you guys are going to take your tubes over to him, and he's
1 north and 8 north.
Let's go ahead and do that now.
And then there's that 6.
Hey, Ben, you're not even wearing jeans. So how do you
Alright, so we can hold it. So we'll walk this way just a little bit more and make sure that everybody's getting in here.
What goes on here? March experience?
Nitrogen.
Yep.
Okay. So
You have a
here at Stanford Research, we exist in this building. This building, when it was first put together, it was actually designed to make computer parts. It turns out that in order to do biological research, you need a building that's not quite as clean as what you would use to build computer parts, which is kind of an interesting idea. So we purchased this building in 2009, roughly, and then we were able to renovate and make it available for the biological research that we do. So this space here is about 60,000 square feet. You guys can imagine your gymnasium at home at your school. Most gymnasiums are around 20,000 square feet. So again, this space is about 3 times the size of your gym in your school. You'll notice that it's only about half full of resources, right? We've got all these shelves and that sort of thing over here. This space is fairly open. We have some room to expand. Again, when Stanford was planning on building their first research building, they had $60,000 or $60 million set aside to build a building. That $60 million would have only gotten them around the 20,000 square feet that we talked about with the gymnasium. So we were able to purchase this building. It's got a lot more space than what we would have had if we were building. If you guys look around, you can see lots of liquid nitrogen. What's the purpose of storing things in liquid nitrogen?
Cold.
It's cold.
Okay, what's good about keeping things cold?
They don't overheat.
Don't denature.
Okay, how? What's denaturing?
This one is like everything. It's when the tertiary structure kind of breaks off and you're only left with the peptide bond.
So what we're looking for is it slows down enzymatic activity, right? So all around your body and in your cells and those sorts of things, there's always different little bits and pieces that are meant to destroy foreign DNA, RNA, proteins that are misfolded, those sorts of things. And so if you freeze cells, especially at the 273, whatever liquid nitrogen ends up being at, Celsius, it stops all enzymatic activity. So cells are not going to grow, they're not going to divide, they're not going to destroy the DNA and the proteins that make up those cells. So you guys will notice that we've got a lot of space for liquid nitrogen here. People normally store their cells. In theory, those cells can be stored for indefinitely. The idea is some of the first cryogenically stored cells were frozen in the 1950s, and in theory, if they were thawed out, you'd still have viable cells. Still be able to. You guys might notice this feature over here. We have a group called SAB. I guess biotherapeutics is the other part of their title. And what they do is they create cows that have immune systems that are identical to humans. So all of the parts of the cow immune system is removed from the genome of the embryos, and then they put in a fake chromosome, a man-made chromosome that has parts of the human immune system. And then when that embryo grows into a cow, all of its immune system parts are similar to human immune system. And so they can introduce diseases to those cows. All of the antibodies that the cows make will be human antibodies, and they can use those for treating diseases. Their treatment strategy doesn't sound, it's not, I don't understand it very well, I guess is what I should say. Again, they work, but they make these antibodies which help us cure and treat diseases. Yeah. So any questions about the facility so far? No.
Do you guys plan to expand?
Do we plan to expand? Stanford Research hires, it's like 2 to 5 PIs a year. Okay, so a PI is a primary investigator. That's somebody that has done their undergraduate degree, they've done some sort of graduate degree, like a PhD. They've also done some amount of postdoc work, which is time spent learning skills after you've already gotten your PhD. So we hire 3 to 5 of those people, and then each of those people is going to have several workers within their lab. That might be something like 1 additional tech, or they might have a staff scientist who has 5 or 6 people that work underneath them. So these, again, when we talk about expanding, Stanford research is sort of rare as far as the field of biology. There aren't very many places that can hire that many people year after year. So we do continue to expand, but we have to expand at a controlled rate so that we can make sure that we've got the resources that we need in order to keep those people working. Yeah. Any other questions? Okay, what do they work on? In there we've got, let's see here, homology.
This battery drains so fast.
They're going to work on kidney disease. So right
But you
now
gotta think
they're
that the batteries-
probably processing some sort of plastic.
Did you bring a charger?
So they're making
Yeah, I brought my
DNA
notebook.
that they want to insert
I
into
need
cells
to plug this
for the mice directly. And
I do have
they've
USB
got this
Type-C,
DNA
but
worked up
it's
in
more-
the
it's
lab.
more
They've
for
got
the-
copies of it, and they're going to be able to put it into cells. Often we use viral transduction in order to get DNA into cells. Just like something like the HIV virus might be used to put DNA into your body, we use certain types of viruses that will accept our DNA, and then they will reproduce that DNA in the cells or integrate it into the gene. So that's most likely what they're doing right now is taking a bit of DNA and making it ready to put into cells. Yeah. So we'll walk around, we'll take a look at a couple of other labs. Or lab sites.